Welcome to another episode of High Alchemy. Today we have Dr. Veera Rajagopal, a physician scientist at Regeneron, one of the foremost scientific communicators in my mind on genetics, society, genetic associations, and he's actually in the Nucleus office today. So thank you, Veera, so much for being here.
Excited to be here. Hi, I'm Veera Rajagopal. I'm a research scientist at Regeneron Pharmaceuticals in New York. So basically, I'm trained as a physician. Then I moved to a full-time research career. I'm doing research in human genetics, and my focus area is to how to use human genetics research to extract insights to identify drug targets. So basically, using human genetics to do drug discovery and drug development.
So Veera, thank you so much again. Great to see you.
Great to see you again.
Yeah, I know this is not the first time you've been at the office, right?
Yeah, this was second time, but yeah, we've been, in touch for a long time, right?
touch,
Yeah,
and I think
even before the Nucleus.
Yeah, exactly. I remember reading GWAS stories way back when. I was one of the earliest subscribers.
We were one of the first person to actually pledge in for a subscription for my Substack, and
Listen, I gotta support the creators. I saw someone who had a passion.
thank you for your support.
Absolutely. I love, whenever I see like a kind of artist, if you will, just like putting their love and their work into something, I just feel like, okay, I have to I have to do something. Like, what can I do? And at that point, by the way, we hadn't actually raised, I think, really any money.
I have no clue about Nucleus or anything. I just, I think I just know you through Twitter, and you've been one of the followers in Substack and Twitter, and yeah, really appreciated your following my stuff and reading about it, reading it.
Absolutely. It's been, and things have developed dramatically since then. So,
yeah.
We have a lot to talk about today.
You're always in the news, in the media. I can see you every day when I open LinkedIn or Twitter. At least once I see some post related to you. You've been trying to convince people to get to do Nucleus genomics tests.
Well, we have a great marketing team. So, I'm just one of many people out through the team company. So let's get started, Veera. I know you have quite an interesting background. So I want to ask you first there, right? I mean, obviously everyone's a product ultimately of where they come from. So do you want to talk more about, growing up, how you became interested in science and genetics?
Yeah, so I'm born and grew up in India. So I'm from South India, near Chennai. So I went to med school in India. So after my med school, so I had interest in the genomics field, in the genetics field, through, like I was like very interested in a particular subject called biochemistry in the non-clinical field, and I was like really fascinated about genetics, and so I took a like specialization. I did an MD in biochemistry, but at that point I have no idea about research or anything. It's just I just loved the concept of genetics and molecular biology. So so I did the MD in biochemistry. But when like fortunately, when for the thesis project, I got a project where I had to do some genetics project in a pharmacogenetics project in schizophrenia of closamine response, and that is when I started reading about it. So that's my first exposure to research, overall research, and also human genetics. I was like really fascinated about the genetics of schizophrenia and also the genetics of brain diseases and the complication, like in terms of like how challenging it is, and we know like really nothing about it. There is like a large, there's so much puzzles to solve. So that kind of fascinated me, and I thought I was like like I thought that's like a really fascinating career to take. So I decided to do become a full-time scientist. So I was like looking
You
for
were MD and then you became a scientist? Or
I did MBBS. That's like a basic medical degree that's equivalent to MD in US. So that's like 6 years. And then I did a specialization in 3 years in biochemistry, like it's lab medicine. And then I worked as assistant professor in the medical college there. And that's when I was like looking for opportunities to do a PhD. Then, I got this contact in Aarhus University in Denmark through one of my senior colleague. And I got this opportunity to visit their lab for 3 months to do a visiting fellowship. And I really enjoyed that experience. And they also liked me very much, and they supported me on an application for a PhD. So I applied for it. I got a full-time, like a scholarship. So I did my PhD in psychiatric genetics. I was studying about the genetics of cognitive, function in individuals with psychiatric disorders, particularly children, with ADHD and autism. Then I continued as a postdoc there for 2 more years. And by the end of my PhD, I was like, I was been using Twitter for I think since 2010 or maybe, but I started using Twitter as a tool as like a way to communicate science, like maybe in the second year of my PhD or something. And
What year is that?
um, should be 2016. I started my PhD in 2016.
Yeah.
Yeah, but I was more active by the end of my PhD, 2018, 2019, completed my PhD in 2019. Then, like I saw the kind of response, like, so two things. One is like, I saw Twitter as a way to connect to people. So, before social media time, the only way you can actually, talk to scientists or connect to people to do network is you go to conference and like even there, you do a presentation. So you don't get that much opportunities to do presentation. So when you do presentation, people learn about your work and you grow your network. But social media was like really interesting because there you get, it's not like someone is giving you, you have to wait for an opportunity. You can just go there and tell the world what like if you think you understood something, explain it to them. And that's kind of thing that I really, kind of got into it because I kind of realized very early that I'm able to reach out to people. I'm able to grow my network. So I started doing it on a regular basis. I really, used to spend a lot of time. I read papers and I don't like pretend or, like overblow stuff, but it's just I kind of use Twitter as like a note-taking system. Every day when I read some paper, I just write it down, 2 points. So I don't like really aim to summarize the whole paper. I don't really, like that's like a huge work. But when I read a paper, something stand out. Like sometimes it be very trivial, it's like people don't even care about it, but it's something that's what stood out for me and I kind of highlight it. But but I kind of realized that people liked it, people kind of appreciated it, like the small things that I highlight from the paper. And that's how my network grew. And my interest like became, broader. Like initially was focused on psychiatric disorders, in neurology field also. But then I was like really fascinated by other fields like metabolism, also in like a lot of, like autoimmune immunology also recently. But I started to read a lot of genetics papers on that, and my network grew, and I got this opportunity like in Regeneron. I
So
think like
Regeneron actually came through GWAS stories. Is that true?
it's not through GWAS stories, but Twitter, like
Just
this
for the
was
generally just like posting every day, and then did someone reach out to you or?
Yeah, so my manager at Regeneron, Giovanni Coppola, he is the head of neuroscience translation genetics group there. He joined Regeneron like maybe just a couple of years before I joined, and he was recruited from UCLA. He is a neurologist, and he has been running a lab there for a long time. So Regeneron recruited him to set up a neurology therapeutic area group, and he's been reaching out to a lot of people. And apparently he's been following me in Twitter, and he learned about my work. And so my Twitter presence has gave this opportunity, for me to. So then one day I got an email from him that he said, I've been following your work. I think you'll be a good fit for a team I'm building. And then that's how I got this opportunity.
Wow.
And then I moved to, like I moved to New York in 2021. It was at still the pandemic is there. So I have got this like special, like a permission because of I was also involved in this COVID project, related project. Then I had to move from Denmark to New York. Then I worked there. So since then I've been working there. And it's really amazing. Like it's completely different experience working in industry compared to working in academia. I've been enjoying so much, been learned so much, and really
What are the biggest differences?
like a lot of people skills that we don't learn in academia, like how to get people to work on it. And often like you make sure that the project there are multiple people responsible for it. So it's like um, how do you say it? Like, if the project succeeds, everyone gets the credit. If the project fails, everyone, like, has to be blamed for, right? So it's not like one person is responsible. Often in academia, it's like just one person. That's usually the PhD student or the postdoc. So I kind of like that because there is an ultimate goal for the company to succeed on a particular project, to find, because the company has been investing on it, but that's not like, so these kind of differences, it's very different. So bottom line is, you are not really trained. So you don't learn the skills when you're in academia, but when you move to industry, you kind of learn a completely different set of skills.
Do you feel that when you moved to industry, there was sort of a, you know, from my experience, and obviously I never did formal PhD or the like, but Nucleus began very much with deep investigation. And then I find that when you start a company like Nucleus, it goes from deep investigation, which is a very specific, unique, focused skill set, to enterprisiness, which is a completely different skill set. And the difficult thing is doing both, because there's somewhat of a paradox, right? Like, if someone is sufficiently investigative, they probably become a scientist. If someone's sufficiently enterprising and not investigative at all, maybe they get an MBA or something, right? But there's always like that sweet spot in between. I'm curious from your perspective, like, do you ever feel when you're, at Regeneron, you're like, damn, like, I kind of miss just doing research for research purposes, right? Because ultimately there was always an end goal now of some sort of drug discovery, right? And it takes, it's a different, there's a different fundamental energy to it, right? How do you think about that? Because I definitely feel that way with Nucleus, where, Nucleus began as my research project, right? And it's definitely something that now, as I build the business, it requires a fundamentally different skill set and perspective. And, obviously I love my job, I love building the company, but if there's anything I miss, it's going extremely deep on something just for my curiosity's sake, just for the insatiableness of it, right? I don't really have that as much anymore. I can't go just pick up, GWAS and read it. I have to, read your notes or the like. And so how do you think about that, as someone who has a PhD, who has a scientist, and then went into industry? How do you think about that?
That's a very good like a point that you raised, because often one of the things when you go and talk to people, obviously I also went and talked to people before I moved to industry, was trying to ask, like, their opinions. Is it, should I take it, or like, because one of the things that people raise is like, you lose your independence. In academia, you can do whatever you want. You can do whatever project you want. It's a curiosity-driven research, but industry, it's like often you have certain goals that you need to achieve. It is true to a certain extent, but it is not to the extent that people, often like believe it so. Because things you can do, or like, I don't think there's a limit to that. I mean, like, you kind of, for example, being in your neurology field, I kind of focus on this area, but there's so many things you can do. And I think, like, in general, people have the independence to explore. So it's not like you have very specific tasks, and often, people have their own things to do.
So you
So you got to figure it yourself, right?
So it is still sort of investigative. You still feel like you're doing, like, kind of satisfying that curiosity, and it isn't just like,
Yeah. So
like, do you ever feel maybe that it's almost as if, you're just doing research for a drug, and it almost somehow limits it? Do you ever feel that way?
no, never. Like, because my passion is human genetics, and what I do right now in my job is to use human genetics for drug discovery.
Yeah.
But the process is just, you use human genetics to understand the disease process, try read about the biology. I'm really always fascinated by how often you know about the disease, but you had no clue about what is the biology behind the disease. And you start looking at the genetics, then it comes out, oh, this gene, this pathway is involved. And these are all process towards the end goal of finding a drug target. But these are very similar to what we do in academia. Like, you are more than independent, like, you are very independent to explore this area. All this, you can do all this kind of exploration, curiosity driven exploration. I think, like, people don't restrict you. I think it applies to everywhere. As long as, like you kind of restrict yourself within specific areas, then I think you can still, satisfy your curiosity in different ways. There's so much to explore. So, so like, so that's even though, like there's some extent, it's true what people believe, that you don't have that much of independence in a industry setting, research setting, but it's not like that much, like you still have, you'll enjoy, and there's no big difference.
So would your advice be to some postdoc out there saying, should I go into industry in the kind of human genetics field? You would say yes.
Absolutely. I mean, like, even though, like, even if someone don't want to, pursue a career in industry, if you want to really learn a completely different set of skills that will make you a better leader, better academic scientist, you should just go there and explore at least for like a couple of years. Then you can go back to academia, and you will go there with a completely different perspective, and that will be like, really, you'll really do well than just sitting in academia for all the time.
And I definitely think with industry too is there's almost this fundamental, trade-off almost where scaling something and productizing something, right, bringing something to the marketplace enables you a level of scale that inherent to the more individualistic and investigative work of academia can never get you, right? Like you could study genetics for 15,
Yeah.
20, 30 years, but fundamentally, and you would contribute to the world's knowledge, which is amazing. But fundamentally, the moment you think, okay, I actually want to scale up this knowledge, I want to productize this knowledge, you need capital, you need
Exactly.
you need scale.
Exactly.
And I think sometimes very, very, very few people have gone through that process of both, of going extremely deep, productizing, and then saying like, how do I scale science? How do I actually bring science? Because it is, to your point, such a fundamentally different perspective and skill set, etc. And I do think that you're right that especially in the context of human genetics, right, something that is so inextricably applicable. I mean, presumably if someone's doing human genetics, they'd probably be very interested not just in the theory of it and the theory of prediction and mechanism, but in actually bringing it to everyone, right? And so I think it's a discipline that inherently is one that just almost de facto in studying it. It's not a bacterial immune response system, though that did bring CRISPR, which is great, right? But it's something that I think if someone's getting interested, it's so intuitively obvious how to apply it.
Yeah.
Which I think is always a really cool thing about genetics.
Yeah.
But even so, and I mentioned this earlier in our conversation before we started recording, it's like, where are the genetics companies? We all have DNA. There's, whatever, billions of people in the world. Whole genome sequencing's come down from $1 billion to now a couple $100. We know, everyone in the industry knows in 3 years it's going to be, $10 to read a human genome, right? The largest full genome database is, 500,000 people approximately in the UK, right? 23 doesn't exist anymore. What do you think? Where are the genetics companies?
I think like definitely a 23andMe thing was like people had very high expectations. We had like an alternative ending, but I'm sure like genetics still have you will see a rise of a lot of genetic companies, but and 23andMe like kind of companies will still serve as a like a example case or reference where you learn about what things worked, what things didn't work, and so your company Nucleus Genomics is one of the many I would expect that it will come rise, particularly now you have AI, which is kind of automating lots of stuff. Even though people, when we talk about AI and what is the role of AI in drug discovery genomics, people imagine about things like AI is able to identify targets or whatever, but the real places where AI will really play a big role is like they kind of automate all the lot of manual tasks that in there, and and I think like scaling up, like you said, genomics price is coming down, and people are realizing the importance of genetic testing and the sequencing. So the data size is growing bigger and bigger, and still there's a whole lot of things that we haven't explored in the genome, like like a lot of so far been focusing on the protein coding regions. There's whole non-coding regions. And so definitely newer companies will come, but that will use more advanced technologies. It will incorporate AI into it, and you will see more uses of like genomics data in different fields, including drug discovery. So I don't think like we will not see any companies because 23andMe went to, gone into bankruptcy.
Yeah,
But
let's talk about 23 some more because one thing that people always say about 23 is, what are you doing to drug discovery? Right? It's one of these people always say, oh, they should have done the drugs, they should have done the drugs, they should have done the drugs. And maybe someone can have the blessing of hindsight now and say, look, it was microarray data, which, so the audience knows it's a small fraction of someone's DNA, right? Focus on common variants, right? Common variants tend to be of weaker effect, or when I say variants, I mean genetic differences or genetic markers. They tend to be a weaker effect. They tend to almost not really be good for mechanism. Obviously, you're much more of an expert of this than I am, but they tend not to be very good for mechanism, hence drug discovery as well, which is why Regeneron actually did their whole exome database, right? And so, maybe someone could look back and say, well, 20 is microarray data, focus on these common SNPs, it was never going to work for drug discovery. I think actually Stephen Shu, founder of Genomic Prediction, he actually sent a tweet out about this. He told Anne, this is never going to work. And he said, I said it. And, it's very easy to always say that, in retrospect. But I'm curious to know, your expertise obviously is the intersection of genetics and drugs. Do you think it was a fool's man errand to try to use microarray data to come up with potential drug targets? What do you think?
I mean, I don't think like that because it's a learning process, right? So the Human Genome Project was completed in 2003. The Genome-wide Association studies the technology that uses this microarray data to measure 300 to 500,000, 500,000 variants, and you impute it. You basically looking at common variants. That's been started like 2005, 2010. And after the completion of Human Genome Project, there was like really so much excitation, and like really the hopes were very high, like what we can find. And the field didn't have much idea about what to expect, like, so it really people thought that, you go and you do like genotyping, you do a microarray for like 1,000 cases, for example, a schizophrenia or diabetes, and 1,000 controls. You're going to go and find the cause for schizophrenia, like one, single gene that is involved, and then you fix it and you're going to cure it. But that's how the thought process was there. But then after studies, studies after studies, sample size kept increasing. Then they understood what is happening, So we have common variants, the genetic variations that are present in, like in a lot of people, right? So, and those variations individually play very little role, right? So their effects are very, very minuscule.
Which is why, just so the audience understands, we build polygenic models. Because the idea is you don't want to analyze a single common marker, you want to combine them together. Because then you have a much more material prediction on basically someone's disposition for the given phenotype, which can be a disease or trait. Yeah, so please. Yeah.
Yeah, so exactly. So that's when people understood the concept of polygenic diseases, like, of the chronic diseases like diabetes, hypertension, or even schizophrenia. All this is like really a polygenic disease, like many common variants together contribute, like each has very, very minuscule effect size, but together they contribute, increases or decreases the risk of this disease in individuals, right? And so that kind of, like went against the expectation, right? People thought we will find the gene that is involved in schizophrenia, we're going to fix it and cure schizophrenia. That didn't work, right? So because to learn that it took like 10 years or so, and then like one thing they understood is like to identify even this common variants, which of the common variants in the genome are associated with the condition, you would need like sample sizes in like millions, hundreds of thousands. So people started doing microarray in large sizes, and that's where 23andMe really played a big role because of they were able to scale it. But very little people, like, of course, like companies like Regeneron and a lot of other companies have been more, focused on coding variations, rare variations from the beginning. There was always scientists who've been like arguing for, you're not going to find any drug targets based on looking at the common variants. But I would say the end field on its entirety actually, like thought initially after the genome project was completed that, like this microarray technology using genome-wide association studies are going to be revolutionize drug discovery. I wouldn't say it didn't, but we learned a lot, So there were many low-hanging fruits that we identified, but it's really, like then they hit a wall, and so
So is that why you think fundamentally the drug discovery process of 23 didn't work? It was because of the microarray data. So retrospectively, would you say, starting drug discovery based off of 23 data doesn't work? Would you say that?
Yeah, so like 23andMe drug discovery is part of it, a big part of it. But initially it was, about using, genetics data to as like getting customers to do this ancestry analysis, also of this all fun analysis, knowing about your roots and everything, right? I think like as you might know better on this, like the business scheme here is that people are one-time customers, right? So once they did, and then that's it, right? So they are not like recurring customers.
Yeah.
So that thing they didn't realize. I think there was very high hope, and people really started believing in all that business model more than what it was. And then the other major thing that they thought, the 23andMe thought, is that, like we can monetize this data for drug discovery. Absolutely, you can use human genetics for drug discovery, but how much you can do, what you can find with the data, people did not have any idea. It was just, like kind of a lot of hypothesis. And for example, GSK, I think, spent like 400 million to get access to the 23andMe data for, like very specific period of time. I'm not sure like how much they got out of it. Apparently, there are like many targets that went out of it, but I'm not sure like how many of them like really succeeded, how many of them went to the trials. You know, I would say that very few handful. You know, but that's how what you can do with a like with a common variant data or anything. And so the realization that because there is like one major bottleneck there is that when you start with rare variants, right? For example, I identify a mutation in a gene that caused the disease, you know. So I start with the gene. I already know what's the gene. So then I can go and create animal models. I create a knockout model or anything.
Exactly.
And then I keep going, right? With a common variant, you identify a variant because they are common, like, so they are correlated with a whole, set of variants in the genome. And you will have like dozens of genes, often hundreds of genes in that area of the genome where you see a signal. You have no clue which gene is like actually playing a role. You cannot just sit and test one gene at a time. And this was a bottleneck that had that common variant, people with genome-wide associations that you went into. And this was not obvious initially. And and also, the cost when 23andMe was beginning, like when starting, that time microarray was itself expensive, and sequencing was much more expensive. It's
Dramatically more
impossible to
possible.
to do in that scale. Like what they had, like 15 million people. It's impossible to do. But now it's like microarray and sequencing almost the same cost. So
So you're saying that 23andMe did the best they could with the technology they had.
exactly. They did the best they could with the technology they had at that time.
At the time, now the world's different.
Now it's different.
Yeah.
Yeah. So
The future's whole genome.
yeah. So even if you travel back in time, you cannot change. You cannot make a different
You
decision.
cannot change the fundamental
Yeah.
technological
Yeah.
limitations. And I agree with you. I mean, 23 started in 2006. The cost of reading a genome was what, 10 million, I think? A sample.
Yeah.
And don't forget, it's only 6 years after the Human Genome Project. Extremely expensive still. And so I think you're absolutely right. I think you're right on a variety of points. I'm going to add a little bit of color in how I kind of see things, which is one thing people forget is that 23andMe was really an offshoot of Google. It was really an offshoot of the Web 2.0 model. What is the Web 2.0 model? Google, Facebook, The customer is not the customer. The data is, right? Facebook and Google both had the same model, which is people come, you use their services, they gather data, and they sell that data to a third party. If you actually think about it in that framework, 23andMe did the exact same thing. The reason why 23andMe didn't build a recurrent product is not because of a lack of will or has anything to do with genetics, not even because of DNA. They could have built a massive consumer health enterprise around DNA. The reason why they didn't do it is because that's not who the customer was. The customer, in her view, in 23andMe's view, was the drug company. The customer was not actually the customer. The customer was a means to basically, and she's actually on the record for saying this, and the CEO, for donating the data, right? And then they used the data to basically sell to someone else. So 23andMe didn't build a recurrent platform, not because DNA somehow is some sort of inherent, diagnostic test that, somehow is only one time and can only, and it's impossible to build this application, but because that wasn't their strategy. The strategy was to satisfy the drug companies. The strategy was very much the Google, Facebook, or Web 2.0 strategy of using the customer as a means of getting data and then sell to someone else. Now, the problem is, and you point this out, is, for Google and Facebook, that worked great for advertisers. In the context of microarray data, because of the limitations of what the technology could do, inherently, unfortunately, it manifested such that the data actually wasn't all that valuable. And this is that I think always find interesting, which is sometimes people will say in the media and culture today, people will say, oh my God, my data, 23andMe is now collapsed, I'm scared. And I really empathize with the customer there, right? It can be really scary to think, oh my God, my genetic data is on the line. But what a lot of people don't realize is, 23andMe, failed because the data actually wasn't all that useful, right? It's not as if 23andMe failed and now that there's data out in the world, they failed because that data actually is not much there. Because it's such a small fraction or fragment of someone's DNA, the most important hereditary markers are just completely missed. And I think finding that culturally, I think now, through Nucleus, you're seeing a change where people are starting to understand ancestry in 23andMe for what it was, which is a genealogy business, and a business principally not about serving the consumer, but a business principally about serving the pharma company. So a company like Nucleus takes a completely different approach, right? We focus on whole genome, right? And this entire dichotomy of rare versus common variants, it collapses, right? There's no common genetics and polygenetic scores versus hereditary testing, which geneticists tend to fall into this. There's none of that. It's you do rare markers, you do common markers. You want to give the best disease risk assessment, right? That's what you're really interested in. And, the one thing that's interesting about, common variants that we talk about quite a bit is prediction, in order to predict, all you need is an association. You don't need causality. In drug discovery, you need some sort of mechanism. I mean, you know this better than I do, right? Which is why I think in that context, especially, whole genome sequencing is even more powerful, right? And not just, exome sequencing. Exome sequencing is great, but even beyond that, right? A lot of people don't realize too, the genome is, the genes are 2-3% of the genome. I mean, it's a small fraction. Most of the DNA actually is not genes. It's not actually protein coded, right? And so when I think about that, and I tie that back to this kind of misinheritability problem, right? And now I'm jumping on genomics concepts here. I hope the audience can keep up, but I know Veera can, which is, hey, we have this problem in genetics, and the problem is that, we make predictions, and some of the predictions are really, really good, right? You talk about schizophrenia, very genetic conditions. The polygenic models are quite good. Height, obviously, everyone knows height is a very good model. But basically, scientists have an estimate of the theoretical maximum contribution that genetics can have on a phenotype. So in other words, if I ask a random person, hey, how genetic do you think height is? They're going to say, it's very genetic. Scientists estimate it to be around 80%. If I said, how genetic do you think skin cancer is? They're going to intuitively say, well, much less, because it depends on the sun more than your DNA, right? Or extroversion, right? Extroversion is obviously dramatically less genetic than height. And so for any given trait or disease, someone should think about it as, oh, well, there's some amount that the genetics explain, right? And when scientists today build models, there's a gap between what the models can predict and what you would theoretically estimate the genetic model could predict. There's a gap. That gap is called misinheritability. Okay, one thing I was going to ask you about is, we touched on AI, but it's all the rage these days. AI. Keon, AI genetics. Is that real? AI genetics? What do you think?
I don't know what AI genetics is. I mean, like, AI is everywhere. Like, artificial intelligence is everywhere. It will be everywhere, and so it's gonna replace lots of stuff, right? So, for example, right now, what all the areas it's been useful for it's like replacing all the time-consuming work. I mean, for now, previously we used to sit and code pipelines and everything, but AI is just helping to do them, like very quickly because of this copilot and everything, right? So that's gonna save time. And then, previously it's really a big hassle to harmonize medical records, right? So one of the important bottleneck of doing genomics or doing association studies is you have to like really mine a massive amount of phenotype database that you get from, like a questionnaire or, like electronic health records, and you have to make sense out of it, put them in a structured way, and then you do association testing. And I think AI is going to revolutionize how you process this data.
So you're really saying that AI's functional role is going to be in taking the existing workflows and making them, instead of something that costs a lot of money, a lot of people, a lot of time, basically immediate.
That is one that's a one use case where really it's making, like a big difference. Then, like this LLM models, they kind of understand everything, right? So the literature, so previously you have to sit and go through the literature. I read every day, like now it's very easy because like the way the LLMs are digging into the literature and getting, like all the relevant studies, that is also like really super useful. And the way if I want to find a gene, I want to summarize the function of the gene, what is known about this gene previously, which might take for me like, many days to go and sit and go through the articles. AI can just do it very fastly and it's useful, but still we go and cross check it. And often, like you might have like, if you go on a topic, you have like 100 articles or 50 articles, you can just feed those articles into the AI, and then you ask questions. So it's literally, like you are searching inside those articles, but not exactly searching for words or sentences, but you are searching for thoughts and ideas, and which AI can do it. So
Amazing.
I have this idea of using this approach to tackle this disease. And is it possible? Literally, without this kind of AI, you have to sit and go through these articles, contemplate on it, and try to think it. But the AI can actually go through it, and it can point to you in the right direction. And these things are revolutionizing how we approach, like drug discoveries and, like, but people often think in different ways, like AI can directly go and point to, okay, schizophrenia, you go and look at this gene, you're going to fix this gene. That's not how AI works, but it's going to
Let's talk about
change
that more,
things.
right? Regeneron has 500,000 whole exome files, right? So, you can imagine in people's minds, there's these rare genetic markers. Do you take this data, map it to phenotypic data? Hey, these people have this disease. Feed it into an LLM. Like literally speaking, because you're talking a lot about the process of doing science.
Yeah, I mean, like,
Actual drug discovery. I mean, have you guys tried it? I mean,
like, so LLMs in general, in the people in the industries, I don't want to talk specifically for Regeneron, but in general, in all the drug industries, they're starting to use it, but not like the way, like you think, like you have all these results, you're feeding into the LLM. Because one thing is like people are really trying to be very cautious in how they use it, because you don't know how much the privacy is. So, they are just testing the waters, including Regeneron, just trying to, incorporate in this specific aspects, how it is useful, but no one has gone, full blown into AI.
though, do you think though, as an industry, you'll take these, not even Regeneron, just think generally, let's say some entity has 1 million genomes. Let's say it's well documented with medical records or phenotypic data. To your knowledge, has anyone taken that, given to LLM and said, come up with the genetic marker that drives whatever X phenotype? Has that happened?
I mean, I'm sure there are startups and companies who are, like trying out these ideas, but it's based on what we learned so far, it's much more complicated. And the level of, you know, p- processing that requires to get those kind of insights is not something, can be easily done by AI, at least as of now. But, things might change, like, this artificial intelligence is really getting very good. But at this time, I wouldn't say like if you do that, you would gonna find all the duck targets. No, it's not gonna work.
Do you think that you'll be able to identify relevant rare genetic markers for phenotypes? LLMs. I mean, we can always talk about like other deep learning models too, which I'm sure that has been applied in the drug discovery context. I know it has been, but how do you think about that, right? Like, I always wondered, like, if you give an LLM like, 100, files, you said these 50 people have this rare thing, these 50 people don't, could it, find the, marker? I mean, you need a lot more data,
See,
sample size,
I'm
size problem, but
I'm not an expert in I'm not an expert in artificial intelligence, but my understanding is that if you're going to do this, then there should be precedent for that, right? Um, there should have been already data in the literature in the world, data world, that people have done it, and there is like kind of concepts that this is how it is supposed to be when you have this kind of information, this is the outcome, that kind of, so that it has to be in the training data for you to feed in this new data into the models to get a meaningful output. But we don't have that yet. We are learning it, so it doesn't know what to look for because it's all based on prior human knowledge. The vast amount of human knowledge that is generated is what is driving this LLMs, right? So if it is completely new to this ecosystem, then I don't think it will only hallucinate. It will try to give you something, but I don't think it will be useful.
yeah, I would think, theoretically maybe it can, look at all the GWASs that were done for a specific, disease, have some sort of intuitive understanding.
I mean, for example, the one area where really AI might be very useful is, for example, like when we get patient in the clinical genetics, you get patient, you do a sequencing, you try to identify what genetic cause for the condition. You have all the symptoms, you monitor the patient, you feed in all the symptoms, you feed in the genomic data, and then you try to identify or prioritize list of mutations. That's like LLM is going to really excel in that area because this is a systematic way of doing, and there is like huge amount of data. There's like thousands and thousands of cases where how people solved genetic diagnosis. Like they use the symptoms, they use this test. So those data is going to help the AI to accurately come up with a good list of variants to test for. So it's going to be really useful because there is a precedent for that, because people have been doing that successfully in the past years, right? So that's AI is going to replace it easily. Similarly, how it happens in radiology, people has been looking at X-ray images of lung or whatever for decades, and they have been documenting it, different use cases, all possible scenarios, and now AI easily will replace radiologists for sure because it's pretty accurate what it is diagnosing with the images, and that's because of the training data. And it's same with the genomics, but only if we already have a systematic way of doing it.
Do you think that LLMs will help with solving misinheritability?
I don't know.
We don't know. Okay. It's the frontier of the frontier. I mean, I don't know
No,
if anyone knows.
I don't think so.
We don't think so?
Yeah.
Last question for you, Veera. What do people get wrong about genomics, genomic drug discovery? What are the things?
Yeah. So I often talk about this particular concept. Like when people, when public, for them, if you go and tell them about, I'm going to do genetics for drug discovery, like not even public, even if you go to doctors or even scientists, often they get this wrong. Is that when you do genetics for like finding a drug, often they think that you're going to study a patient, right, a group of patients, and they're going to do a genetic testing or going to do sequencing. Let's say there's some patients with diabetes and controls, then often people assume what you are doing is like you are trying to find out what mutations of them are actually making them sick, and you go and try to fix that mutation, and so you're finding a cure for the disease, which is true in some cases, like rare disease cases, but that is not actually like what exactly people are doing in drug discovery. So the way like human genetics is used in drug discovery is like like it's human genetics is like what we call it as like it gives you data from nature's experiment, natural experiments, what we call it. So when you do a clinical trial, let's say you have a drug that inhibit a protein, right? So you have a drug that inhibits a protein called PCSK9, and when it inhibits this protein, the cholesterol level comes down. So you are going to prove it using a clinical trial. You take 10 patients and then give them the actual drug, and then you take 10 patients, you give them like a placebo, you don't give them any drug, and then you test them, you give them the drug, and then you monitor them, and then you see their blood cholesterol levels goes down, right? And to do this, there is a lot of risk. It's very expensive to do clinical trials, and there's some things can go wrong. You get like a lot of severe adverse effects. People can die. But what if there is a way you can actually do, like you can, there's already an experiment that's been done. We can use that experiment to get some insights, confidence before you go to the clinical trial. So that's the human genetics, right? So I go and look into the protein, the genetic sequence of the gene that encodes the PCSK9, and I try to identify, like mutations that reduces the PCSK9 in the blood. So I compare people who have the mutations who don't have the mutations. So these people have 50% less PCSK9. So I look into their medical records, I look into their life history and ask the question like, have they got heart disease in their lifetime, or what is their blood cholesterol level? And I get evidence that suggests that if you reduce this protein, you get, like you see this result. So then that becomes very helpful in, like to kind of conduct the clinical trial. So genetics becomes like a natural experiment. So it kind of gives you, like a kind of a human trial that you actually don't want to conduct. So that's why we go and look into the whole genome. We try to identify all these kind of, natural experiments, these mutations. Often we know what we are looking for. So for example, we have a drug already in the pipeline. We go and try to find individuals who have mutations in related to this particular drug target that we are looking for. We use this data to de-risk our pipeline, but often we go and find a completely new genetic gene that has been associated with the disease. Then we go and start from there, because we already know from the data that if the individuals who don't have a functioning gene in their body actually is doing well, they are protected from heart disease, they're protected from liver disease, then we go and start looking into it. So it kind of points you in the right direction. So it's not always we are trying to go and try to fix whatever the genetic, mutation that's causing the disease, but we are using them as a kind of instrument to help us to identify targets. So that's the beauty of using human genetics for drug discovery.
That's amazing. No, that's a great answer. One final question is, do you have any questions for me?
So what's new about Nucleus Genomics?
I would say, I gave you this answer earlier, and I'll say this to close the podcast, which is sometimes when people think about a genetics company, they'll say, oh, it's a consumer business like 23andMe Ancestry, it's like a genealogy thing, or a gimmick, or it's a clinical business like Natera or Myriad, and it's like serious and it's medical. Or maybe it's a preconception testing business and it's for couples or families. Or maybe it's a pharmacogenomics business, right? Pharmacogenomics is how your DNA affects metabolism of drugs. Maybe that's what you do. Or maybe you're in IVF testing, genetic testing business. And people treat each of these things as its own thing and its own companies and almost their own industries within the broader genomics industry. I would say Nucleus is all of them.
Okay.
We're the end game of genetics. That's what I'd say, Veera. That's what's new.
That's super exciting. I have one follow-up question. So we've been focusing a lot on drug discovery, and we also explored the case of 23andMe, who, like had a huge expectations on using the data for drug discovery, right? So my question to you is like, are you going to get into drug discovery at all at any point in the future for Nucleus?
I think that the first thing I'd say to this is that we really believe as a business in giving consumers complete control and agency over their data, right? So by default, we don't share any genetic information or protected health information with any third party, right? We're bound by HIPAA. We give, people can download their data at any time. We really want to give people control. Now, people do come to us and say, hey, my father had Alzheimer's. Can you use my data to help find a cure for Alzheimer's or a treatment? And in that context, for those patients who explicitly opt in and understand that, hey, my de-identified data will go to a company for helping come up with a new drug to try to save my grandfather, my mother, my sister, whoever, absolutely. I think for those patients who explicitly opt in, I think consumer genetics and drug discovery got a really bad rap. And it's not because I think actually people, think that drug discovery is not important. I think anyone intuitively, when they've gone through a loved one or themselves that has a disease, you're willing to do anything to try to help cure that disease or at least, slow its progression. The problem was trust and transparency, right? People felt as if they were sort of duped, right? They did this data and then, oh, the data is, in GSK's hands. And, I'm sure you probably know some of the scientists at GSK. I'm sure they're very earnest. 23andMe, I think the biggest problem was not necessarily in the philosophy of trying to, continue to push medicine forward, but was in the communications and ultimately not treating the customer as what they should be treated as, which is the fundamental owner of their genetic information. So that's kind of the way we think about Nucleus.
Okay, customers come first.
100%.
Okay, that's amazing.
Absolutely.
Things move in the pace that we see right now. Like we had no clue this AI or something like AI is gonna come like few years ago, and suddenly there's this chat GPT and everyone is going crazy about it. So like these kind of breakthroughs is happening every day. Every day we are hearing about it. So if this is how it's gonna be, then surely it's gonna be like 10 years or 20 years from now, we're gonna see that kind of situation. And I'm sure Nucleus is gonna play a big role in that world.
Yeah, I think so. I think you're right that 20 years, definitely. Human genome on every smartphone. Ubiquitous. And I do think Nucleus is going to play a strong role in that. We're going to help do that. 50 years, I think I'm going to do a 50 to 100 year prediction, which is, I think one thing people underestimate about genetics is that there's a genetic stack forming. Sequencing DNA, analyzing DNA, understanding DNA, and engineering DNA. All these technologies are going to come together. And of course, AI for me is a tool. AI is maybe the most powerful levered tool ever created, but a tool ultimately in of itself is not very useful. It's useful to do something, right? And I think AI is going to be most consequential in human genetics because ultimately there's no question as fundamental as what does it mean to be human? That is the most, I think, fundamental question. And I think you alluded to this earlier in our conversation, but I think it's not communicated enough. We know so little still about the genome.
Yeah.
We know so freaking little. It is the way I want people to think about it. The intuition is it's almost like the moon or something, right? Which is like, yeah, we've been there, we've walked around, we have some specimens, we can tell you things about it. But there's no one on the moon, right? It's like the molecular equivalent. It's still this frontier for us to understand and to explore and to really figure out, right? And the most fundamental questions just of the galaxy, of the celestial world, of is there other life, that equivalent is true in the molecular level too. From the molecular to the celestial, it's the same thing, which is we don't know if there's life. And similarly, out there, we also similarly don't understand life in here. And so there's something about that I think is really, really profound that, people don't yet see. I think as a business, for me, Nucleus is my baby. It is the reflection of my own soul. And when I think about Nucleus, I zoom out, I don't think about it in the next 1 year, 5 years, 10 years. I think about it in 100 years. And I think that in 100 years, you will see a revolution of humanity. And what does it mean to be human? And I think a lot of companies will say, okay, if our technology works, or if the thing that we want to do works, it will change what people do or how people do something. I think Nucleus as a business is asking a much more fundamental question, which is, what does it mean to be human?
When you think about a company like, doing such a big like with big aims, like doing its Nucleus, like a Nucleus Genomics, doing genetic testing, talking big things like, like treating, changing the healthcare or predicting, disease risk, you would expect someone who has been in this field, who was expert in the field, but you wouldn't expect like this kind of ideas, this kind of, forget about the ideas, the belief, you believe in this concept or this system, or someone who is like, who has just who hasn't that much gone into this field, right? So like Kian, and it's really, like fascinating to see how someone strongly believes in that and just goes, like full into this business, right? Trying to create a company based on this kind of strong belief in the value of genomics. And I don't know how that happens, how people, what makes someone to really, inspire someone to do that. And it's like really amazing. And often based on my experience, that those are the people who often succeed. And, like, because there's something that motivates them, that they don't back out. They don't, like, there's like kind of a very pathological drive that's pushed them to do things, even though, like they there's a lot of things go against like against them. So that's like really amazing to see founders like that. So
Thank you, Veera. Thank you so much.
thank you.
It's been great chatting.
Good luck.